Welcome to my channel and welcome to Recombinant DNA Technology. Here in this video, I am going to discuss how to solve restriction mapping problems in 5 easy steps. But before I enter into the main topic, I would like to say something about restriction enzymes and how restriction mapping is performed in the laboratory. Restriction enzyme is a type of endonuclease that cut the double-stranded DNA within it if a particular sequence is present within the DNA, and the sequence is called recognition sequence or restriction sites. They produce same fragments each and every time, and that's why they are very useful tools in RDT. They are also used to map gene and chromosome. In the laboratory, DNA is first digested with more than one restriction enzymes separately and in combination. When they are used in combination, it's called double digestion. And after the digestion is complete, the digested products are separated using gel electrophoresis. And from the size of the fragments, the map is generated. Now, I am going to discuss how to solve restriction mapping problem in your examination very easily using these 5 steps. Step number 1. Find out whether the DNA is linear or circular. If the DNA is linear, then the number of fragments will be n plus 1, where n is the recognition sequence. Here in this picture, a linear DNA contains only one recognition site, so the number of fragments are 2. And in the second case, there are 2 restriction sites, so the number of fragments are 3. But in case of circular DNA, if only one restriction site is there, then the number of fragments will be 1. And if 2 restriction sites are there, then the number of fragments will be 2. So, if you know how many fragments are produced after the digestion, you can easily calculate how many cutting sites are there. I have already told you that after the digestion is complete, the fragments are separated using gel electrophoresis. But if 2 fragments are in same size, they are not generally resolved in this technique. In that case, you may miss some fragments. They are called masked DNA or missing DNA. To know how many fragments are missing and what is their size, just follow this rule. Size of the original DNA minus total size of the fragments. For example, in this picture, a 12 kb linear DNA is digested with 2 restriction enzymes. They are SMA1 and second one is NOT1. In case of SMA1, 2 fragments appeared. They are 10 kb and 2 kb. So 10 plus 2, that is 12 kb. That is the actual size of the original DNA. But when the DNA is digested with NOT1, only one fragment appeared. In this case, 6 kb. So total size of the original DNA, that is 12 kb, and size of the fragment, that is 6 kb. So 12 minus 6, 6 kb is missing in this case. So in NOT1, only one fragment appeared in this gel, but the number of fragments are 2, 6 plus 6. At first, compare the fragments appeared in single digestion and double digestion. You will find that some are common and some of them are unique. For example, I have compared fragments between EcoR1 and double digestion. 7.4, 6.2 and 2.9 kb fragments are present in double digestion. That means these 3 fragments are not digested by BAMH1, but 8 kb and 4.5 kb fragments are missing in double digestion. That means these 2 fragments are further digested by BAMH1 and produce some small fragments. Your task is to find out which fragments arises from 8 kb and from 4.5 kb fragments. Here in this example, 8 kb is digested into 6 plus 2, these 2 fragments, and from 4.5 kb, 1 kb and 3.5 kb fragments are generated. In this way, you have to find out the possible combination for the 2 fragments that appeared in BAMH1 digestion. The 2 fragments are 12.9 and 10.1 in this case. So from 12.9, 3.5, 7.4 and 2 kb fragments appeared, and from 10.1, three fragments appeared and they are 2.9, 6.2 and 1 kb. In the step number 4, you have to align all the fragments according to the correct order you have found in the previous slide, and after the alignment the map looks like this. And in the final step, draw a line and place all the fragments according to correct order. And finally, point out the cutting site or the recognition sites for the two enzymes. Your map is complete. So, in a nutshell, step number 1, find out whether the DNA is linear or circular. Step number 2, find out any masked DNA present or not. Step number 3, find out the possible fragment combinations in double digestion that derived from the single digested products. Step number 4, align all the fragments according to partial similarity. Step number 5, draw a line or circle and place all the fragments according to order you found. Problem number 1, a 14 kb circular DNA is digested with BAMH1 and KPN1, and the data are given in the table. From these data, you have to construct the map. At first, DNA is circular, and in all three cases, the total size of the fragments are 14 kb, so we are not missing any fragments in this case. And in BAMH1, the three fragments are produced, and they are 5.1, 5.4 and 3.5 kb. 5.1 kb fragment is further digested into 1.9 and 3.2 kb fragment, and 5.4 kb fragment is further digested into 4.6 and 0.8. 3.5 kb fragment is further digested into 1 and 2.5 kb. And in KPN1, three fragments are produced. They are 6.5, 1.8 and 5.7 kb. 6.5 kb fragment is further digested by BAMH1 into 1.9 and 4.6 kb. And 1.8 kb fragment is further digested by BAMH1 into 0.8 and 1. And 5.7 kb fragment is digested into 3.2 and 2.5. And when I have aligned all 6 fragments side by side according to partial similarity, the map looks like this. And from the alignment, I have constructed the map, and the map looks like this. Problem number 2, a linear DNA that is digested with EQR1 and BAMH1, and from the data, you have to construct the map. Here is the solution. The DNA is linear and the total size of the fragments are 16.5 kb in all three cases, single digestion as well as in double digestion, so we are not missing any fragment. The fragment 8.5 kb and 5 kb generated in EQR1, and the possible combinations are 2.5 and 6 for 8.5 kb, 4 and 1 for 5 kb, and 9.5 kb fragment produced in BAMH1, and the possible combination 4, 3 and 2.5. And when I have aligned all the fragments side by side according to the partial similarity, the map looks like this. So from the alignment, here is the map. If you like my video, don't forget to like, you can share, you can subscribe and you can comment. And thanks for watching.